Good morning to all. So, I welcome you all for a third session of today's international conference. So, I'm happy and proud to introduce today's speaker, Dr. Ashwin Kumar, who is currently working as an assistant professor in the Department of Biomedical Engineering, SRM University, Katangulathur. He completed his undergraduate in Biomedical Instrumentation Engineering, followed by MTech in Nanotechnology from Amrita Center for Nanoscience and Molecular Medicines. Later, he earned his PhD degree in Biomedical Engineering from IIT Madras. His field of research include nanoformulations as contrast agents for cancer prognosis, design of nanomedicine, development of low-cost microfluidics chips for biomedical sensor applications. He has been awarded DST MHRD scholarship for pursuing postgraduate degree studies. He is a recipient of various international travel grants, even abroad presenting work in scientific conferences, including to visit to Japan. He has been awarded Best Oral Presentation at International Conference on Bioengineering. He has also received a graduate research fellowship during his PhD degree at IIT Madras. Dr. Ashwin is currently working on 3 different projects, namely low-cost optical imaging systems, development of high-throughput delivery of solutions, and hybrid nano-contrast agents for preclinical imaging systems. So, with this note, I welcome Dr. Ashwin. Thank you.
Hello. I thank Dr. Mohan Loganathan sir for introducing me, and I also thank Dr. Mohana and the chairpersons of Dhanalakshmi University who have invited me for this talk. And it's been a day. You all have a great lectures over the 2 days, and I'm just going to start with some basics of what we are going to do and what is this ANS. ANS is Advanced Nanotheranostics Research Group that we have at SRM Institute of Science and Technology in Katangalathur. And today's topic is on molecular synthesis of lab on a chip. I mean, we are going to see how lab on chip technology has already been talked by Dr. Ashish, the previous lecture. So he has been discussing about how this lab on chip is going to help in several biosensors or basically the blood plasma separation. But what we are going to look at in this lecture is that how we can make nanomaterials in lab on chip. So that is what we are going to look at. So let's see the outline of this talk. Is first we'll be discussing what is the basics of nanotechnology. I'm not going to talk more details on how we manufacture in a bulk, but we are going to see how these nanomaterials can be developed in microfluidic devices. So how the lab on chip and what is its physics behind in developing this or synthesizing these nanomaterials in lab on chip technologies, and how we can have different materials like say metal nanoparticles like say gold, and we have metal oxides, iron oxide nanoparticles, and also different quantum dots, and how these can be generated in a chip. Because we see whenever someone talks about nanotechnology, right? So we always know that we take a beaker and we start circulating a magnetic bead in the beaker, and then we develop this nanomaterials depending on the temperature, pressures, and we give certain conditions to develop these nanomaterials, right? So that is what we generally use when we make or when we fabricate certain structures of materials. But how these nanostructures can be grown into a microfluidic device? So that's going to be an amazing thing, right? So first I'll give you a brief introduction of nanotechnology, then I'll say how we can translate into an microfluidic devices. So this is my group. We work on several aspects. One is a low-cost microfluidic devices. That's the base that we'll be discussing soon. And we also work on imaging agents, basically a contrast, wherein we can try to image a cancer using an X-ray or MRI or optical imaging systems. So we're also developing a low-cost medical instrumentations in our departments. So as a biomedical engineer background, so we also incorporate making this low-cost device structures, low-cost devices that can also help in translating the research for a working. Yeah. Okay. Yeah. Thank you. So, I mean, this is a roadmap of what we have been doing. I mean, what I have been doing later years. So imaging agents, and we develop low-cost medical devices, and we do a lot of hypothermia aspects. We also work on osteoarthritis, and we also work on cancer hypothermia. And also we develop all this, low-cost devices for liquid biopsy samples. So before we go into what is the role of nanoparticles, we have to know why we have to develop these kinds of nanomaterials. We talk about cancer. There are different ways we can see a lot of hallmarks that can have these cancers which can grow much prevalently, right? But the problem is the drug translation, or we identify not all the drugs is going to work for all the patients. So that's going to be one of the major challenge. So what is a major problem in a cancer is that we can see a lot of hallmarks that is present in a cancer that makes the patient or the drug that is not making relevant to the patient, which is not working for a cancer patients, right? So we have seen breast cancer patients, we have seen colon cancer, lung cancer, we have different cancers. But all these cancers have a different hallmarks, and these hallmarks are definitely going to play a major role on identifying the drug, whether it's going to work on a patient or not, right? So in order to understand this, we cannot go directly to humans. So we need to work on a preclinical, which means animals or rats or rodents, whatever that we do. But for that, even that, we need to have a lot of challenges. We cannot use a general clinical imaging systems that will facilitate identifying how the drug is going to interact with the cancer, right? So in this, we'll be developing a lot of contrast agents that can work to identify how a drug is interacting with the cancer tissues, right? So it is quite important to have, you make something in the bench, which means in a lab, that has to be translated to a clinic. So there is a lot of clinical testing, preclinical testing is going on. So this is all very important when we go for an understanding the interaction between the drug and the cancers, right? So it is quite important. Now we have seen a lot of tremendous technology have come up with, development of these nanomaterials that makes it more significant in understanding about the interaction of this drug and the cancer. So talking about the fundamentals of nanotechnology, we know the different particles. As you take a bulk, so this is going to be a bulk material. So you take 1 meter by 1 meter, right? So you start breaking down. For instance, you have birthday cake. You start cutting it into different structures, right? The whole volume is same, but the surface area is going to be high, right? Can you see the same block is cut into 4. The volume is same, but what happens to the surface area? The surface area is higher, right? Similarly, I'm trying to cut down into much more details. I'm cutting into 2, and now I'm cutting into, like, say, 4. So what is happening to the surface area? It's obviously increasing, right? But again, the volume is same. I'm not changing the volume. So this is what a basic fundamental of nanotechnology, how a nanoparticle is going to work. We say, no, the nanoparticle is more fascinating. We say the optical properties are different. Later we'll see about those things. But what makes it unique is that you improve the surface area. You consider this is going to be a brick, single, We talk about a brick. You take a brick and put it in a bucket of water. What happens to the brick? When the water gets loaded into the brick, right? The water diffuses, we say. You break the same, after you take it from the bucket and you break it in the center, and you see only a small area is getting interacted. In the center, the water will not go in. So what's happening there? Because the surface area is less, right? Similarly, if I start cutting into 4 pieces, if I start putting down into a bucket, what happens? Again, the water is more, right? Because of the surface area that is increasing, the interaction between the environment and this materials. So that's what makes it unique. So that's what we say, nanoparticles are very reactive, right? Nanoparticles are more reactive, and it acts much faster when compared to a larger bulk surface. So that is where the importance of nanotechnology coming here. We talk about the interaction, we talk about the how this material is going to talk to a cancer cell or to a human environment or a biological environment, right? This is going to play a major role. So we are going to cut down into pieces, and we make how small that we can create. So that will facilitate in how much amount of interaction that can create to a biological environment. So we have seen different types of nanostructures. If you see from here, we have organic, we have polymeric nanoparticles, and now we brought a bulk into smaller particles. And these are all different particles, but the materials are different. We are not using only a polymeric material, we also use a metal, we also use a metal oxide, right? So we have different kinds of materials dropping down from 100 to 1 nanometer. That's how we define what is a nanotechnology. So if someone asks what is nanotechnology, your answer should be the size of having a particle. It can be a metal, or it can be a metal oxide, or it can be a polymeric. If it's size in the range of 1 to 100, which is actually the size of, that's how we describe nanotechnologies, which is basically a science of small. That's why I kept my lab name as ants, right? Ant is small, right? So we have science of small that we are going to talk about, right? So we have two different approaches. How do we make this? Sir, it's quite interesting. Yes, we are making nanoparticles or different sizes. You say 1 to 100 nanometer. How do you make it, right? So when you make it, we have two different approaches. So I said, you take a big brick and you break it down and you make it into particles, right? This is one way. Or the other way around, you take all this small chemicals, the molecules. You start taking all these atoms or molecules, and then you start building into a nanostructures. So ultimately, we are going to make this nanoparticles, whether it's going to be a top-down approach or it's going to be a bottom-up approach. In a very simple terms, if I want to say, if you go to kitchen, you make a biryani, right? What happens? You add all these things into a pressure cooker, and then you make it into the aroma of biryani, right? You feel tasty as the time is 12 o'clock, so everybody feels so hungry, right? So this is what, I would say nanotechnology it's not new, it starts from the kitchen. Whatever that you see, whatever that you are cooking in your home is what we are trying to bring out into a nanostructures. It's maybe very comic, but this is the reality. Because when you want to make any food, you need to know what is the concentrations of the materials that you're going to add. For instance, salt or sugar. And then what is the temperature you're going to cook? What is the pressure that has to be maintained? How many whistles that has to come? So all these will be, maintained when you want to make nanostructures, right? So this is what general ways of preparing nanostructures. There are different types of properties when you make it into small. It's not when you use it in a bulk. When you make it into small, we have different properties that we can attain. So what kind of properties are which? So if you see, we can talk about the difference in the size. We can make, say, 1 to 100 nanometers, and we can have different particle sizes. We can increase the surface area, and we can also increase the composition. If you want to make different materials to come in, that is also possible, right? So we have different kinds of properties that can be generated depending on your manufacturing or fabrication of these materials, right? So material is quite fascinating when we make it from a small to a larger particles, or breaking down from a larger structure into a smaller nanostructures. So how this nanoparticles can be prepared? So I said, as a biryani is going to take an example, you have all these agents. You add salts, the water, and then you add the masalas, the ingredients, and then the rice, right? So this is what the similar process is going to take place. So wherein the first is, this is what the Lamer theory that he talks about, what is happening when a nanomaterial is going to synthesize, right? You add all these different concentrations of your materials that you want to develop. Say, for instance, I want to develop a gold, so I need to have a gold precursor. If you don't have a gold precursor, I cannot make a gold, right? So if I start adding my gold precursors, At a certain concentration, what happens is it forms a nucleation. So consider how do we relate to an analogy is that you take a glass of water and you start adding a pinch of salt. What will happen? It will solubilize, right? If I'm adding one more spoon of, like say I'll make it more, in a mathematical expression, I'd say I'm adding 1 gram of salt in 1 liter of water. It will dissolve. I'm adding 2 grams of salt, I'm trying to dissolve. Okay, it got dissolved. I'm adding 5 grams of, which is more than the water can dissolve the salt, which is not going to happen. What is happening there? You find there is a sedimentation of the salt, but it is the super saturation that's happening within when you try to dissolve this salt in the water, and that super saturation is what the first stage of the formation of nanoparticles. It is the addition of atoms and you make into a bulk structure. I mean, a few atoms, if I take like say 100 atoms, it becomes say 1 angstrom. If I take 1000 atoms, it becomes 1 nanometer, right? So this is what the super saturation, the critical super saturation point after which it starts growing. So depending on your concentration, you are increasing the salt values and you're slowly making the particles bigger and bigger and bigger, right? So this, what happens? I need to stop somewhere because I said nanotechnology works only from 1 to 100 nanometers, no? I cannot go more than 100, so I need to stop somewhere. So this growth will start taking place depending on the amount of concentration of salts that you're going to add to the salt that you're adding to the water. So I'm going to stop this. How am I going to stop this between 1 to 100? How am I going to tune it? By just adding some surfactants. Surfactants is what? Having some kind of a stopping materials that will not allow the excess of other atoms that can come and bind on your structure of, on the surface of your nanoparticles. So that is where I'm stopping. That is what we call it as surface modifications, or we call it as an agents that will help in stopping of growth. So I need to know at what point I need to stop this growth. So that is my aim, right? Say I want to say I have 10 nanometers. I need to know, okay, I should stop here. If you want, say 50 nanometer, I should stop at this point. If you want like say 1 micron, then I think I need to add more. But again, the size is also depending on the amount of concentration that you add, the precursors that you add. It doesn't stop there, right? You need to know how much amount of concentration is to be added so that we can have or we can stop the amount of size of particles here. So this is how it works. So we talk the metal precursors, a gold. I take some reducing agent, I start forming these nanoparticles, and then I stop this coalescence. See, the metal precursors, slowly it's becoming into a particle. So now, a molecule that convert into a nanostructures, and this nanostructure will stop by either modifying or changing the concentration, or by changing the surfactants, or adding some surfactants to it. So we have different methods of batch reactors. We say chemical synthesis, physical synthesis, and value. I've worked most on this synthesis protocols because we develop different structures and different nanoparticles that will also facilitate in making this unique nanoparticles for certain applications, like say imaging agents, or I want to make it into iron oxide nanoparticles or whatever it is, right? So this all different methods wherein you use a laser-based ablation here. To ablate a material and you make structures. As I said, you take a bigger stone and you break it down. So that's what is going to happen in this method. Wherein the chemical synthesis, you're going to take precursors and they're going to make it into a nanostructures, right? So ultimate aim is to make nanoparticle synthesis depending on our need and depending on the size, depending on the properties that we require, depending on the applications. So all these will come into picture when you want to choose which kind of modality or method that you want to identify or to make nanostructures. So in our lab, we use a lot of these. When you go and look into a chemistry lab, you find all these instruments, right? We have this furnace because I need to give a heating. I need to have a stirrer because I need to mix it. I need to have a sonicator to break it. Or I need to have a centrifuge to separate it. So what do you think if I start incorporating all these devices into one small chip? Do you think it is really possible? So we have this big lab with all these instruments. So if I can convert into a small, I would like say 2 inch by 2 inch. I'm making it a little more bigger, but. So do you think it is possible to bring this much big lab into a small device? Can you make nanomaterials? Let's see. So here a new paradigm of developing nanoparticles or nanomaterials that is coming in. So here we have this microfluidic devices. We call, that's what Dr. Professor Ashish was talking about, the interaction of bulk surface aquastofluidics into microfluidics to do a blood plasma separation. But here we are focused on how to generate nanostructures when you use this microfluidic devices. So microfluidic devices generally used in different applications. How do we incorporate this nanochemistry and where do we apply this nanochemistry into different biological applications? Because as I said, we need to increase or we need to fasten the clinical translation when you wanted to make a drug that can really work on a patient. So that is where we need to make it much faster. The problem is the batch, well, I'll tell you what are the problems of the batch reactors. There are so much of, problems we talk about different properties there, the size, the structures, the composition. So it's all going to come to play a major role. So if you want to control all these things, that is not possible in a beaker. So I want to bring out into a smaller micro reactors that I can make a beautiful nanostructures that I wanted to create, which will explicitly work on certain diagnosis or certain sensing. So how do we understand this microfluidics from nature? So this is an important thing. We see two different aspects here. One is a big tree. You have this, xylem and phloem you would have studied in your 12th standard, right? You can see a lot of, you pour water in the bottom of the tree and the water goes up to the top of the tree. How it actually goes? That's a fascinating thing, right? So that is what the microfluidics is going to play here. We have these two structures. We would have studied this in 12th standard, the xylem and phloem, which is going to be the transportation of the water and the food, right? So if I pour water here, the water can travel across till the leaves at the top. And similarly, if you see, there are the rivers or, the lakes that can actually go and, reach the ocean. You have a lot of water flowing through this. When you start interacting or understanding these two phenomenon into microfluidics, you can understand two different concepts. One is a laminar flow, another one is a turbulence flow. This is what is happening. So I'm just making a smaller structures. I'm making a microfluidic channels wherein I can see two different important, I would say the major phenomena that we could see, a microfluidic flow that can happen in a channel is one is a laminar, another one is a turbulence flow. Right? You pour water on you wash a car, right? You see the water that can pass through a pipe. The water is passing through the pipe. The phenomena is going to be different at the ends and also at the surface. If you say water is traveling across the pipe, in the center you have a free flow of water, but at the ends or at the ends of the pipe, you will have always a resistance. And that is what a turbulence flow is. Again, laminar flow, and this is turbulence flow, wherein you bend the pipe, what happens? There is a turbulence flow. Just understand this to identify what is, or to really understand what microfluidics is working. Right? If you understand these two concepts, microfluidics is very easy. So as I said, we have all these bigger instruments coming into picture. So now I'm going to use a very few equipments, I would say a very low cost equipments that can eventually make my nanoparticles at a specific structures. Say for instance, I said nucleation, growth and formation is what my idea, right? I'm going to make a metal nanoparticles or metal oxide. So what is happening is I'm going to flow whatever the precursors that I'm going to add. Here I'm controlling the flow by a syringe pumps. Syringe pump is a device wherein you can infuse the flow of fluids in the channels, in the micro channels, We can see whether, how much amount of concentration has to be given. Here I'm trying to tune the concentration only by when you use syringe pumps from externally. So what I do is I connected small tubing from a syringe pump that can connect it to a beautiful device with different designs of structures. I'll also tell you how to fabricate a design and what are the design consideration we need to have. So if I start doing it, initially I have a nucleation that's forming here and growth is forming here, and slowly it's getting extended and you form a bigger particles. Now I'm giving a small molecules, or molecules, precursor materials that that travels across these through channels, and the laminar flow and the turbulence flow will definitely help in making a small diffusion that's happening within the fluids, and it creates a beautiful nanostructures in this regime. Right now you see there are 3 different structures, and all are getting connected into a purple color, and then it's converted into a gray, which means there is a formation of something. Right? So this is how the bulk reaction is going to convert into a microfluidic structures. So henceforth, you want to develop some nanoparticles, definitely think about the microfluidic devices that we can make in our labs that can facilitate a specific size and specific structures of nanoparticles that we can develop. So these are some of the differences that we could see between the batch and the lab-on-chip devices that we can make these nanostructures. So here we have, like say, batch. It's going to be like say every time you need to take a beaker and then you need to stir it, you need to add, and you have the time is it is a problem with the amount of concentration you're going to add in the beaker or the size of the beaker. Either of the things will be problematic for us. Say for instance, someone would have developed a particle size, gold nanoparticles of 10 nanometer in a 100 ml beaker. If I ask him to do it in a 1 liter of beaker, there might be a problem. Or if I want to scale up from say 1 milligram, I want to make it like say 10 milligram. Again, you need to increase the concentration. There is a problem again when you want to make these nanoparticles, because you cannot play with nanoparticles just like making a biryani from smaller pressure cooker to a bigger box, right? It's not possible. So there is always a lot of factors that involves here, and that can be overcome, and that challenge can be converted. It can make it more better when you bring into a lab-on-chip technologies, right? Everybody would have shown nanostructures that can be developed in a beaker, but if we can bring this into a microfluidic device, because I'm going to give all my fluids in an external environment, and that can travel across the channel, and it's going to be a continuous method, and I can develop all these nanostructures, beautiful structures. You want to say only in metal nanoparticles, you want to make metal oxide on top of this gold, you want to make quantum dots. We'll see some examples that we have done in our lab, and also what we are doing from other labs as well. So some of the key considerations we need to have is that one is you need to know what are the precursors that you need. You need to know the basic synthesis and what is the design, how you want to make this, how much the flow rate has to be given. All these has to be considered, and for what application we're going to use. So these are the 3 major considerations we need to look. So a basic microfluidic device will have some inlets, and you have an in outlet, and you have some reaction chamber. And this can be converted, this can be used for making this nanostructures. And what are the different applications? I would say the microfluidic based nanoparticle synthesis can have advantages over the conventional batch reactor is that you can have a control in synthesis of these nanostructures. And second one is you can decrease the cost and energy time, and also requires less time. Say for instance, you consider a biologist who wanted to make gold nanoparticles. You cannot go and ask him to give a reaction and state that, okay, you can use this reaction, you can make some nanoparticles. Rather, what I would do is, I give him my chip. Hello ma'am, you can use this chip. You have this flow rates, you on it and you go. When he comes back, after an hour, she gets a beautiful nanostructures, gold nanoparticles, whatever the color that she wants. Because as we know, gold is not yellow in color. Bulk it is yellow, but when you break down into nanostructures, the colors are going to be different. Whatever color that we want, we can make it from gold nanoparticles. The only thing is you need to tune the size, tune the shape. That's the only 2 parameters that is required. Right? So don't give a beaker and gold chloride solutions to make gold nanoparticles. Rather, give them a device, ask them to develop. So you make their life more easier rather than asking them to understand the reaction. They don't need to understand your reaction, biologist, because we have been talking about, we have several doctors, they don't ask you, see, you prepare me material, last week you gave one size of material and today you give another size of material. And this is not going to work and the previous one was working, give me the previous one. And we don't have the control over the reactions. And this control over the reaction can just come only by using a microfluidic devices. So you give them device, you ask them to make, and then they are happy and you make the patients happy, right? So these are the different considerations or three different parameters that we can see. One is a continuous laminar flow, another segmented laminar flow, another droplet based laminar flow. The plus and minus is what the pros and cons of each each microfluidic devices or the designs with which we use. So continuous, generally they use it for polymeric nanostructures. So you want to make some polymers, like say you want to make some chitin nanoparticles, you want to make some PLA nanoparticles, PLGA, because all these nanoparticles is a very good applications in drug delivery applications, right? So if you want to use a continuous laminar flow, you can create a polymer structures. But there are few instances you need to go for segmented flow and droplet flow based method, because this droplet flow based method wherein you can create droplets here. So can you see here? So these droplets is going to be your beaker. Don't consider as a droplet, consider as going to be a beaker. Only thing is it is going to travel in a channel, micron channel, right? So if rather than having a bulk big instruments that is having a 100 ml beaker and then stirring it, I make everything into micro droplets, and now I can also control the amount of reagents that I need to make that droplet into the channel. Make it easier, right? We can find it much easier, no? So next time we don't need to think, because when we synthesize nanostructures, we always try to understand what is going to happen in the beaker. I always try to do that. What would be, how these chemicals are getting interacted, how these nanostructures are forming, how these atoms are coming up. I mean, that's a fascinating thing, but you don't need to worry now. So now we can start tuning it. If we understand one droplet structure, what is happening in the chemistry, we know the whole system is going to happen in the same way. And understand every droplet is going to have the same amount of concentrations, because our flow rates that is giving through the inlets is same. So every droplet will have same amount of concentrations of precursors that we are going to add. So every droplet is a beaker for us. See, now we make hundreds and hundreds of beaker in a channel. Amazing, because we are going to make nanostructures. I am making micro droplets here. It's easier. I am trying to control my reaction in a chip. Beautiful, right? We can make these things. The droplets would really make the specific amount of concentrations that can be added within the droplet is going to be same across all the droplets. Now you can ask me, sir, you're going to have a gold chloride, and now I am adding a precursor material, a reducing agent. So now I want to add another material into the droplet. Is it possible? Yes, that is also possible if I have another inlet that can exactly fuse on this droplets. So fusion of droplets can also happen within the channel. Then I can make how much amount of concentration I need to add to this droplet. So now I can tune my droplet depending on the flow rates, depending on the number of channels. I'm increasing from two inlets to 4 inlets. So I can do it. I can make it more, better reaction into the droplet, and I can create a beautiful nanostructures. Right. So we'll see both this continuous laminar flow and the droplet-based laminar flow to make the structures, right? Different structures. So what are the design considerations that we need to go? If you look at, So we'll focus on most on this droplet-based mechanism because most of the metal nanoparticles or metal oxide nanoparticles or quantum dots, we generally use a droplet-based mechanism that can provide you a beautiful structures, I mean nanostructures, whatever the the monodispersed particle that we can create. Okay, so what is that happening is that the droplet generation is controlled by the formation and deformation of the liquid-liquid interface between the two immiscible liquids. So how these droplets are going to generate? So definitely I need to have one aqueous solution and one oil. So you take oil and you put water into it. What happens? The surface tension happens and then droplets form within the oil, right? So this is happening just because of the surface tension, because it's going to be two different liquids. Similarly, I'm going to have two different liquids. One is aqueous, another one is an oil. So when the water comes into the oil and due to the surface tension, and it creates a droplet. That's how the mechanism is going to take place. Very simple. We don't want to make it complicated. I want to make a beaker, micro droplet beaker. How I can make is that you have an oil and you start infusing this aqueous solution, and depending on the flow rate, you can identify or you can change the droplet sizes. Right? So this will make a highly parallelized experiment, and you can create a monodispersed particle sizes, and it can facilitate the control over the droplet size, and we also have a small sample volume. We don't need to, say for instance, I change the syringe pump flow rate. Definitely I can also change my particle sizes. If I increase the concentration, I need to just change the parameter of the syringe pump flow rate. That's going to, that's external that I'm going to add. That's all. So I can do everything depending on my need. Say for 100 droplets, I need spherical structures. Next 100 droplet, I need a star structure. Next 100 droplets, I need a rod structures. Impossible. The only thing is your syringe pump has to tune in such a way that the concentration of the precursors is going to be different. Very simple as that. So how these droplets are going to form? There is one mathematical, I'm not going to go more details on this mathematics because I could find a lot of UG students and PG students here, so I'll talk little overview of what this droplet formation is going to happen. So here, one thing is the capillary number. The capillary number determines the droplet diameter decreases as the capillary number increases. So I'm not going to go detail, but what is exactly happening here is, say this is what the aqueous is going to come in, this is where the oil is going to come in. So I'm changing the flow rate. So what the first thing will happen is, there are three different things that can happen. One is a squeezing. So it will either, because of the flow rate that's going to happen, it will squeeze or it will push the water, and then it forms this droplet. Right? Next one is the dripping. So if I start increasing the flow rate of my oil, what happens is the squeezing will not take more, because here you could see a large amount of squeezing here, but here it will be very short and it starts breaking down much faster, and you make it a smaller droplet here. And here what I'm doing is I'm just trying to control my reaction rate, the flow of rate. I'm increasing the aqueous flow rate. What happens is, again, there is a jetting process takes place. Either of the things can happen when aqueous and oil interacts. So this all basically depends on the capillary number. So how do we design this microfluidic droplet formation is that we have varieties of designs that can be created. Sir, is that only one design? You showed only one design. The previous was only one design. It's just a T-junction. You have oil and you have water that can interact with this, and it starts forming a droplets. But this is not going to happen. We can create droplets in several geometries. See, everywhere we can create droplets, but look at the geometries are different. Geometries are different. The droplet sizes can also be tuned. That's a fantastic thing, right? So you see a flow focusing geometry wherein you have all this, the flow that's going to pass through it, and then it forms all these droplets here, and you have co-flow geometry here, and you have, because what happens is this oil will squeeze here and it forms droplets here. The squeezing or jetting, whatever it is, it's going to happen at this stage, because you have enormous amount of flow that's going to come in, and this flow is also coming from either sides, and that will break your water, and that forms a droplet. So similarly, a core flow focusing, there are several geometries that can people have designed and created to generate these droplets, and we can also modify the amount of precursors that we want to have. Say here we have two different colors. You say I want to make some iron oxide over a gold. I can do it. So red is an gold and green is an iron oxide, iron chloride. So I can create a Janus particles. So anything is possible. So depending on your requirement, that's what I said. What type of nanoparticles you require, whether it's a core shell nanoparticles or just only metal or metal oxide nanoparticles or quantum dots, depending on what we want is what we identify this design. Right, so droplet can be formed in any ways, but this choice of the design is basically depends on your particle that you require. So different fabrication techniques are used. We have basically this microfluidics or the lab on chip technology we talk about from the timeline states from say 1900s to till now. So there have been several ways with which you fabricate this microstructures, microfluidic devices or lab on chip technologies. So basically is we have this glass and silicon as the first primarily they have developed, and second one is the PMMA, and we have this paper base. Now we have so many, if you go to even I've been to SRM Medical College, they have so much of testing that happen within the papers. So for instance, urine analysis, they're identifying there is a strip which has a different sheets of papers, and all these papers identify the different components of a urine sample. So that's amazing, right? So all these paper-based microfluidics are also coming in recently so that we can make a low-cost devices rather than having, when you make a silicon or glass, you need to have a very high clean room, and also you need to have sophisticated equipment and well-trained people. Affordability is again a big problem, because again the output of the developed structures will also be higher, right? So here we have another one is a polymer-based structures, and I am working on this PMMA, acrylic-based devices. We have been fabricating these microfluidic devices, but again there is also challenging in both these things. This could be an alternative for glass and silicon, but when you want to go for much smaller dimensions, and this is more viable when compared to this. So you need to choose according to the applications. I find most of the diagnostics that can happen within the blood, which doesn't require a much smaller channels. So I can use this polymethamethacrylate that can be used, PMMA based devices that can be eventually helpful for identifying several blood-based diagnostics. So I'm trying to identify, I'm not saying this is bad, but I'm saying this could be an alternative for what application I'm looking at. Because I want to work only on blood-based diagnostics, obviously I'll find this is more better. I don't need a clean room here. I just need a very few instruments which can make it up to a price of, like say, one device. I can make it like for 200 rupees or 100 rupees depending on what is that. So these are all the ways with which since we are not much exposed to all these silicon based, we don't have much of facilities here, but we are trying to bring out which can be done in my lab itself. So we are developing these polymeric, PMMA based microfluidic devices that can provide beautiful structures, whatever the designs that you want, and we can create your nanostructures. So these are the different design configurations when you wanted to make. This is the first generation. If you see, there are different generations. You have one inlet, one outlet, and this one inlet and you have one outlet, but you have a serpentine structure. So what's the big deal in having these two designs? Do you think there's going to be a change? Yes, it's going to be a change wherein you want to mix it. So how do you mix it? So for instance, you pour two water in a channel or in a pipe. At the end, it will be the same if it's going to be a straight tube. For instance, you bend the pipe, and then you flow the two channels, two liquids into it. At the out, you find a different color. And this mixing will happen only when you have these structures. Because a rapid mixing will take place at these points. At these points. So what happens is, the two liquids will flow through this. Because it has to bend, it will hit on the surface of the walls. And again, the diffusion takes place within this U channel. And then it mixes, And that is where our beaker, we are changing the magnetic stirrer with the serpentine structures. Because we have these magnetic stirrers, right? We put magnetic beads into it and we stir it. I don't want to put magnetic bead. I'll add this simple U bend channel, or we call it as a serpentine structures. I mix it. So mixing can happen this way, right? And we have different channels, and now this mixing, the second stage is droplet formation. How this mixing is going to take place? We have, sir, you said this channel, it's fine, it's mixing. But how, if it is going to be a droplet, how it's going to change? You take a ball and you hit it on the wall. What happens? It doesn't come back. I mean, if it's going to be a different structure, it hits there and it goes there. When the ball rotates, it's not going to rotate in the same direction. It will revolve, and it hits on this and it revolves at the other side. So what happens if I put a liquid into it? So again, I'm making a reaction chamber there. So now if I make a droplet that can travel across these serpentine structures, I can make my droplets to mix much more rapidly when compared to a normal fluid that's going to flow through this serpentine structures. Excellent, right? Can make, the droplets, you understand, you in a ball, and I mean, like say, when you go to, this theme parks, you travel like this, right? What is that? I didn't recognize. So what happens is there is a rapid mixing that happens. Some people will get vomit. It's because, there is a mixing that's happening within your body, right? The same thing will also happen within these micro channels, right? And these are the different stages with which you want to mix it. Now we are making much more rapid mixing when you have these structures. Because a ball has to travel this way, it has to hit all the pillars. And you say you have a classroom of 50 people and you want to travel from this point to other point. Obviously you will hit at least some 10 people there. So this is what is happening here. Again, we are having a mixing property here. A single stirring mechanism can be brought out in different structures of microfluidics that we want. This is not only for a nanoparticle synthesis, this also helps in a blood-based diagnostics. Or you wanted to interact a blood, or you want to separate the blood plasma, the WBCs, platelets, and all these can be happening. And another important aspect is the density, the size of the particle is also matters a lot when it passes through this channel. Because you put the blood in Ashish sir was talking about the blood plasma separation. The RBCs goes one side and plasma goes other side. It's just because of the density of the RBC when compared to WBCs. Right? That what makes it separatable when we use it in a channel. So this is what we have been developing over time. I mean, we have some challenges. We have been working for at least, like say, 1 or 2 years on this. A master student who have worked on this, and he had come with a patent. And what we did is we just design our microfluidic channels in a 3D microfluidics. This is an open source software. You can use it when you want to make these structures. And then we started using a laser engraving technique to create channels. And then we start making this 3 channels here. I mean, we have taken a 1 mm acrylic sheets, and then we start making all these micro channels within the thing, right? So the center layer will have the design with which we want to create, and the top channel and the bottom channel will fuse, and the top channel will have all these inlets, and the bottom channel will be the liquid that should not go out of this micro channels, right? So we need to seal it. So we have a different mechanism with which we seal this thing. So first thing is we have understood that the thermal bonding, we call it as thermal bonding. You just need to have these 3 plates and fix it at a very high temperature, almost like say 150 degrees Celsius, because the glass transition temperature of this PMMA is going to be like say 170 or something. So we have it at like say 140, and this glass transition temperature, it makes it to bind these 3 layers, right? And the problem what we saw in this is that thermal bonding is good, but eventually we have understood there is a lot of issues in having a different structures here. See, you can see there is a lot of corrugations here, And these corrugations has happened just because of the heating that is given on this microchannels. Though we have got almost like say 250 to 500 microns, at least one dimension we have gotten, the micron regions. But then we have find there is a lot of problems that's going to take place when a fluid of interaction is going to take place. So we have found there is a lot of problems when we use these kinds of testings, and apparently we are not able to see a transparency one, because PMMA is transparent. I'm not able to see transparent when I use a heating environment here, and we could see a lot of leakages because the heating is not proper, and all these things have been coming into picture when we use this thermal bonding based methods. So what we did is we finally moved on to a solvent based method. So we have created this. This is already been known that UV lamp can be used for creating this binding of these three microfluidic, these three layers of PMMA. But what we did is we had added much more inherent properties, or we have changed the solvents that can exactly modify your surface of this the first three layers, and then we can fix it, and then we got a beautiful structures. I'll show you some. We also optimized these designs. We also changed the different problems, and we also changed the different methods. We used ethanol, and we also used acetic acid, and then finally we have come up with a composition of having this different solvents to create this beautiful microfluidic devices. And this has been filed as a patent, and provisionally, it's still there. So that's why we are very reluctant in giving some informations. So can you see here the droplets have been generating? So it's a T-based junction. So the droplet size is almost like say 500 microns. So now I can make a beaker of size of 500 microns. Right? So now I can add all my reactions there. I can create all my reactions there. It's possible. Another important challenge is in a batch-based reactor is that you have a heating environment. But sir, then how it's going to work in a droplet? You're not giving a heat. You might ask this question. What we do, in a cooker, we add all these things, right? We heat it, we give a pressure, but we are not giving any heating here. The good advantage of having this is that you don't need much of heating, is because you have very few species are getting interacted. The amount of species that is getting interacted is small. So you don't need an external energy to make them into nanostructures. So that's another important thing, right? So that's why all these micro droplets will give you a beautiful structures when you make these droplets in your channel. So what we have done is we have made a very small design in this, and we have created all these droplets here. So we use all the syringe pumps, and finally we could able to get some, the droplet sizes. So we have also tuned, we also understood what are the different lag phases, and these are all how this necking is happening, how the droplet is creating when the two fluids are getting interacted, two immiscible liquids are getting interacted. So here we have 4 different stages are there. One is a lag phase, wherein because the flow is going to go, it will always have some kind of resistance here. It will not come in. So it has to come in because the pressure is always given from the either side. So it starts coming slowly. It's like a thief that's entered into a house. So first, initially he will hesitate to go inside, but later he finds there is no dog, then he'll come in, right? So similarly, the filling stage starts coming in, and then later the necking stage, and finally the detachment. Detachment is breaking down into a droplet and coming out into a channel, right? So we have designed this same microfluidic devices, but this is with the bled droplets. So what we have designed is that we have created some kind of a bled-based droplets, and this droplet will be used as a diagnostic technique, and this is also provisionally filed in, it's provisionally filed. It's not get granted till now. So what we have done is that we have made all these droplets varying the flow rate ratio. You can see that the size of the particles are changes. The droplet sizes are different. Here if you see the flow rate ratio, which is 0.2, the droplet size is almost, say this is the channel size. See how channel is very clear. What you have made the channel, previously you have found there are some surface structures that is present, but now we could see a very smooth structure here. This is all optimized over years. So the flow rate ratio has been varied. We can see the droplet is also different. The flow rate ratio, when I change the flow rate ratio, you can see the channel size is same, but I'm just tuning only the size of this solution. I mean the syringe pump, through the syringe pump. The flow rate is different. I'm able to optimize my droplets here. The droplet size is high here and it's low here depending on my flow rate ratio. So now let's look on how do we develop these nanostructures in the molecular synthesis of gold nanoparticles in a lab-on-chip technique. So here we have seen there are different colors of gold that can be generated, and we know the gold nanoparticles is going to be a most biocompatible material as we call till now. The gold is much tunable and it can easily functionalizable, and you can biocompatible, and you have all this X-ray imaging can be used using these gold nanoparticles, and hypothermia is also used. The photothermal therapy, you can use gold nanostructures of different sizes, and we can tune the hypothermia for cancer treatments. So we have developed some gold nanoparticles. It's a batch reactor. We didn't do it in a microfluidic region. So we have created 10 nanometer particle sizes, and we started injecting in animals. So because we have an in-house X-ray imaging system, we have developed these contrast agents. Can you see here iodine? There's the same concentration. Here the concentration is 350 mg, but the gold nanoparticle is 23 mg per ml. So this concentration of gold is injected in animal, where we are trying to identify the atherosclerosis of rats, right? So that will facilitate whether there is any the blockage that's happening in the heart vessels, and all these things can be imaged using an X-ray CT technique. So this one is developed using a gold, the gold nanostructures have been developed using a lab-on-chip technology. So now you see we have 3 different inlets and 1 outlet. The 3 inlets, we have an ascorbic acid, a growth solution, and a seed solution. So seed is a gold seeds, almost like say 1 nanometers, and you start having a gold chloride, a growth solution, and all these things forms a droplets here. So all these droplets, depending on the concentration of ascorbic acid and depending on the concentration of seed, my particle size are different. Here we can see bony structures. Can you see here a bony kind of a structure? Right here it's going to be like say spherical structures. So I have tuned my inlet parameters wherein I can identify, I can create my structures well. So there are several other examples wherein they have used again 2 different reactants, and they have used continuous channels, and they created beautiful gold nanostructures here. You can see spherical particles of gold that has been created, and these are pretty old experiments that they have created. Now it has been much more increased into a uniform or monodispersed particle structures. So here another example is having a gold core shell nanoparticles. So wherein you have cobalt and gold. So you have gold 3 solutions. So you had all these 3 things into the channels, and then you slowly it forms hollow gold nanoparticles, and then you finally create a gold pegylated structures. So metal oxide nanoparticles is what we have also been developing in our labs. And we know metal oxide has different properties of having gas sensings and also semiconductors. A lot of semiconductors, superconducting materials or super paramagnetic particles can also be used for magnetic hypothermia and also for MR contrast agents. So there are several advantages of having this materials. What we have developed is a pretty good nanoparticles, wherein you can excite these nanoparticles in an X-ray and you get a luminescence. Generally, how we get a luminescence is that you excite an UV rays and you get a luminescence. That's what we have seen till now. But what we are proposing is that we have got something stating that when you have a material which can excite using an X-ray, you get a luminescence out of it. So we made a material wherein you can, it's going to be acting as a contrast agent for animals. So I want to see what is the structure of what is the tumor volume that is present in the animal. I create a tumor environment or I create a tumor model. I want to see what is the size of the tumor. I can see it through an X-ray. But I want to see it through an optical imaging system, it will be a very difficult scenario. So wherein, if I use an X-ray, X-ray can penetrate across your body. So wherein I get a luminescence out of it, and I find all these structures. I identify the molecular functionality. Very simple. So we make a material wherein, so we used different structures. A microemulsion method is a batch rearray that we made. So we are trying to bring this into microfluidic devices. Still we are working on this protocols. So it's a microemulsion method and doesn't require any kind of heating or pressure that is required. It's a simple technique wherein we used a europium doped with the tantalum oxide. So tantalum is a very good CT contrast, and europium is a very good optical agent, wherein even if you excite with UV, europium will also give you a luminescence. But the good point is, when I start doping this into a tantalum oxide material, what I found is extremely different. So can you see here, this is not an optical imaging system, this is an X-ray imaging system. I excited this powder, my powder material, my powder sample, which is exciting using an X-ray. I could see a beautiful red luminescence. So this luminescence changes depending on the voltage that I'm giving here. So I'm increasing my voltage, like say 50 kilovolt, 100 kilovolt. So that's what we do with an X-ray system, right? So we tune the properties, tune the parameters of voltage, current, and exposure level. So depending on this, increasing the voltage, we get beautiful red luminescence. And when you get red luminescence, we can actually overcome different biological interferences, autofluorescence in the human system that can be overcome. So this is what we call it as a scintillating material, or we call it as a zeol material. Zeol is X-ray excited optical luminescence. Generally we see UV excited optical luminescence, but this is going to be an X-ray excited optical luminescence. And very few groups are working across the globe. And we are working on different materials to identify where all this X-rays can be used. For instance, we use one of my PhD scholar who's working on osteoarthritis imaging, where in most of the aspects we use an X-ray imaging there. So you want to understand the molecular functionality. How do we do it? So only when you have, so you need one X-ray source or one source where you can have two different imaging modalities. So one with an optical, another one is an X-ray. So you get both structural information and also you get an optical information. This is how we have this. Oh, 5 minutes, okay, yeah. Okay, so there are different, so people have been talking about the circulating tumor cells. So spions have been used. We use a lot of different diagnosis techniques, and we generate microfluidic devices. And I'll show you one good video. Yeah. So can you see here? These are all iron oxide nanoparticles. We flow through the channel. What's happening here? I keep a magnet here. See the droplet here and see the droplet here, what is happening. I'll just go back and come. Oh, sorry. Can you see here? The droplets are, the magnets are getting attracted. So what I have, if I have another channel here and I bring all these droplets across my channels, it makes it easier for me to isolate. Whatever that you want to make it, you can isolate from the blood. So we are adding all this. These are all the preliminary experiments that we are working on. These are all unpublished data, but still I'm showing it. Wherein we have all this nanoparticles that can be attracted through a small magnet that is present here, and then we can capture whatever the CTCs that we want or exosomes that we want. It's all the magnetic interaction which is going to happen within the channels. It's going to be interesting, or for a better diagnosis, this will be really helpful. And okay, due to the time constraint, I'm just moving at the end. There are few things that I wanted to show you because we can create some affordable products. We'll walk through some exciting science. It is not my work, but I just want to motivate students so that they really wanted to bring out some good science. Don't think always science come from a big lab or big. You need large equipments, or you just need an innovative thought process. You know this, right? What is this? CD drive, DVD drive. What we can do with this? We can write, read. That's what generally people do, no? But there is one person, Professor Anja Bosin. She is from Denmark University. What she did is, she has taken this small device. You know what is exciting about this device, you know? This is the most important part of a CD drive. If you break it down, this is the most important part, which is we call it as an optical pickup unit. There's nothing but you have a laser here. And this laser, the size of the laser that hits on the CD drive is going to be 200 to 500 nanometers. Remember. So if you want to do some biosensing at nanoscale, I can use this. We have been using pretty longer time. But how do you use this optical pickup unit? And there are tremendous works that have been done, not only by this. She's one who started how to use this optical pickup unit. She has been using this as a biosensor, but several people have come up with different aspects. They have developed a FT Raman spectrometer. You won't believe. If you buy a FT Raman spectrometer, it's going to be like almost like say 25 lakhs to 50 lakhs. But can you create it using a one CD drive? That's a question. They have created. One of the things you need to know some electronics and where it is going to apply. Right? And this, I'm not going to go more details of it. You can just look at this paper to get the more information on this. So wherein I want to bring out saying that this FT Raman spectrometer using an optical pickup unit can be used as a diagnostic technique even in hospitals. It's going to be a very low cost. And another one is, you think it's a 3D printing? It's a 3D printing, but the structures are in micron. This is created using an optical pickup unit. This is fantastic, no? So one CD drive can give you all these things. It's not that easy, because if you say stereolithography, SLA technique, wherein they use UV laser to fuse or to make structures, and this Blu-ray player, which has Blu-ray, is actually a blue laser. So this Blu-ray laser can create a structure. Because I said we can create a microstructure just because we have nano-sized things, right? And then using the DVD drives, they've also developed 3-axis stage 3D printers. So I'm just showing you this because you can start making one 3D printer at your home. One thing is you need to have 3-4 DVD players. That's all. It will work. And this guy, Manu Prakash from Stanford, he has developed two different things, which is very simple. One is the folderscope that he has developed, and second one is we used to play in the childhood. This guy has patented this. Do you really believe? He has patented this game. You find it as a game, but he finds it as a science. He's unfolding the science. What he did with this is that he's separating the blood plasma here. That's amazing. Right? You do it for 2 minutes, the blood plasma is getting separated. And the only thing is, where this can be implemented for developing countries like say Africa, a lot of people are struggling to isolate blood plasma. And this guy has developed a very simple thing. You just need to, do like this for a few minutes, the blood plasma gets separated. And he has done I think you need to read this paper because he has done a several amount of, experiments to what is how much RPM that it rotates. You think, it's just a rotation, but it rotates almost like say 5,000 to 6,000 RPM. Sometimes when you even do it more higher, it goes up to 15,000 RPM. That's what he claims. 15,000 RPM at which you can separate it. Ultracentrifugation can happen. So that's all I want to bring you. Is just to bring you an motivation of what science is not. That is always coming from bigger labs or something. Though they are at Stanford. This guy is from Stanford. He could make a very small devices, which is at very low cost, and you can get one folderscope from Flipkart or Amazon. You can get it. It's a paper-based microscope. And I acknowledge all my professors from different universities and my PhD supervisors and my master supervisor. And I acknowledge Dhanlakshmi Srinivasan College to bring me to give a talk. And also I thank all my students who also helped me in doing a lot of experiments, and we are coming up with good science. Anyway, everybody, everybody is more interested in making some science, and science should be more affordable. That's what my thing is. Thank you.
Thank you, Dr. Ashwin. Now, it was a very excellent talk, and he was narrated in a very interesting way with a lot of analogy. Thank you very much. So now the session is open for discussion. If somebody has a question, please raise.
It was just super, sir. The session was a really wonderful session, and the main, I think my students, they were waiting for this particular session where basics are also covered. A few days earlier, we conducted a program on nanotechnology, and we called a resource person from NIT Trichy. Even he also did not go much more detail on basic part. But Dr. Ashwin, I really appreciate him for understanding the situation or the level of the students who are available here. And he started his presentation from basics onwards. Even we all were aware that, okay, nano means 10 to the power minus 9. So this type of information only we were having. But how to create that nanoparticles? There are many other methods, centrifugation methods are there, or even syringe pumps are available in the market. So these basic concepts are being covered, and their applications also were covered very wonderfully by Dr. Ashwin, and I really thank him for understanding our levels, and he has presented in such a manner. Another thing he has given in between the example of biryani. Maybe sometimes he wanted to say that today sometimes Dr. Mohana would have arranged biryani only in lunch. So he has aggravated our hunger also simultaneously. And the only one thing I want to inform to my students, maybe many of the research scholars are available who would be conducting research in this particular area. They would be knowing more, and faculty members are also available. For them also, it may not be some, we can say that surprise. But for our students, I feel that certainly it is there. The information which they have received, it will be really, really very good. I want to quote two sentences said by Thomas Leonard. So I quote, "People are empowered." Please try to listen word by word. People are empowered when they switch over from the perspective, I cannot, to how can I. So there is a need for all of us. It's not that only for the students, that we need to switch over from the perspective, I cannot, to how can I. And then really this type of research work, when they will be doing it, certainly they will be getting very good results. So I unquote that, and I feel that most of our students, they will be switching over from the perspective, I cannot, to how can I. If we will switch over, certainly something different we will be in our later years. So thank you very much for giving me an opportunity, for giving feedback in this particular session, and I really enjoyed the session, sir. I really enjoyed. Special thanks to Dr. Ashwin and Dr. Loknathan and Dr. Mohana and all the participants. Huh? Of course. Thank you very much. Huh? Right. Please.
Thank you, sir. Thank you very much.
Any other questions from students?
Without realizing, we do use a lot of nanotechnology in medicine, especially in the pathology lab. When there is inflammation and blood flows, and this is what happens. You said no microfluids. That happens inside the body. When there is a wound healing, it just separates into small microfluid particles and flows through that small gap made and brings in inflammation. So that is basic pathology for you. And then we apply a lot of this in IHC. immunohistochemistry, nanotechnology. We break that into small particles so that, so that we can use very little reagent, which is very costly. Even one drop is about 32,000, 40,000. So we do use a lot of, maybe you can, in the next talk, you can use all that in diagnostics. Thank you, wonderful, wonderful lecture. Wonderful.
Hi.
Please give him a big hand. You didn't.
And once again, I want to thank him for making me to remember my days when I was taking classes for aeronautical engineering, when he described about laminar flow and turbulent flow. Okay, so actually after coming here, I forgot all those things. It became my past days, but he made me to remember all those flows. And when I was studying, teaching about boundary layer theory. Okay, so this laminar flow and turbulent flow is very much, we can say, when we are doing research work on aerodynamics, when the flow used to be there over the wing, air flow. So that is there. So thank you very much, sir.
Yeah, please go ahead, Rizwan.
Hi sir, this presentation is going to be very useful for interdisciplinary research areas, and thanks for that. And my question is about sensitivity and surface area is very important for nanoparticles. See, this form of synthesis of microfluid nanoparticles compared to other form of synthesis nanoparticles, is there any difference is there for sensitivity and surface?
If your question is, if I understand your question well, you're asking how to tune the particle size, or the surface functionality.
Surface functionality and sensitivity also important for nanoparticles.
Sensitivity in the sense, what kind of sensitivity?
Reaction.
The reaction. The nanoparticles reacting in the.
With others, if we induce that nanoparticle into any human or plants or any bodies, so the reaction sensitivity, react sensitivity.
No, in biological environment that can happen, the interaction can take place through a phagocytosis or pinocytosis, and sometimes it is cavially mediated. So it depends on how well this nanoparticle or the surface functional moieties that you have it on the surface will definitely enhance. And also the zeta potential of the nanoparticle will also help us in interacting your particles into the cell surface. If you drop down close to the plasma membrane and the nanoparticle interaction, so it's going to be a different strategy. Depends on what we want. That's what I said. The sensitivity or the selectivity that we require depending on what kind of particles that we make and how do you want to modify it.
Okay. So from synthesis side there is no difference.
Synthesis side, the difference is that you can have, even you can do surface functionalization within the droplets. That is also possible, and a lot of people have worked on that as well. And definitely, it is you can tune it. You can tune.
Okay,
Yes.
thank you.
Sir, first of all, awesome work, sir. Yesterday I have presented my poster presentation regarding nanotechnology, Respirosite, sir. On that presentation, the target therapeutically finished product, how will be separated, I have reviewed. On that, Respirosites can be filtered by centrifuge, and it will be separated with the plasma, and the plasma will be recombined to form a healthy blood, as I have reviewed. Is that possible?
The possibility is you are asking how we separate nanoparticles from body, human
Sir,
system.
it's still been a hypothetical theory, sir. According to you, is it possible in future?
I'm not getting your question clearly. Maybe you can repeat.
Sir, separation of Respirosite from blood is still been a hypothetical theory. But
The nanoparticles with blood you are talking about.
yes, sir.
Okay.
Artificial red blood cell, Respirosite.
Okay.
It's been that will be separated by centrifugation, and it will be separated by plasma, and blood will be separated to form a healthy blood. It's a hypothetical theory, as I presented yesterday. Is there probability to do that project? Yesterday I have reviewed, sir. That is an hypothetical theory.
Okay.
If that is useful, it will be
See, the usefulness or application is how do we make these nanoparticles that can exactly interact with the blood cells. You say you want to make this respirocytes, right? That's what you have been talking about yesterday, if I really remember. And the nanostructures that you wanted to infuse to make better respirocytes, I think it's possible.
Sir, can I ask in Tamil?
Oh, okay.
Okay. Sir, இந்த ரெஸ்பிரோசைட் பற்றி நான் எடுத்தேன் சார். அதில் வந்து என்னென்னா பிளட்டில் வந்து ஆர்டிஃபிஷியலாக ரெட் பிளட் செல்லை எப்படி வந்து நம்ம வந்து இன்ட்ரடியூஸ் பண்ணலாம் அப்படின்னு சொல்லிட்டு நேற்று வந்து நான் எடுத்தேன். அதில் வந்து அந்த தெரபியூட்டிக் எஃபெக்ட் வந்து அந்த கோலை வந்து அச்சீவ் பண்ணிடுச்சுன்னா அதை வந்து நம்ம வந்து எப்படி இது பண்ணுறோம், திரும்பி எப்படி செப்பரேட் பண்ணலாம் அப்படின்னு சொல்லிட்டு நான் பார்த்தேன்.
Okay, you are talking about isolation.
Yeah, isolation.
Isolation, there are so many techniques are there. See, even if you take industry waste, there have been so much of effluence that comes out. We can use several techniques that can, magnetic based separation is a big big theory, or there are so many things have been there to isolate even ions. It is possible to isolate it.
ஒரு இன்ஸ்ட்ருமெண்ட் நீங்கள் காமிச்சிங்க, அதில் என்னென்னா வந்து சென்ட்ரிஃபியூஜ் வந்து பண்ணலாம், அடுத்து வந்து பிளட்டில் உள்ள நானோ பார்ட்டிகளை வந்து நம்ம வந்து பிளாஸ்மாவை செப்பரேட் பண்ணலாம் அப்படின்னு சொல்லிட்டு நீங்கள் காமிச்சிங்க சார். அதே அந்த ரெஸ்பிரேசைட்டும் ஒரு நானோ பார்ட்டிகல் தான், அதாவது ஒரு நானோ டெக்னாலஜியால் கண்டுபிடிக்கக்கூடியது தான். அதையும் வந்து நம்ம செப்பரேட் பண்ண முடியுமா? அது வந்து 1 micrometer.
If it should have some properties. Say for instance, whether you want to do it using a magnetic things, or you want to target or capture it, and then you need to separate it. So depending on the isolation technique, that will favor. And you should tell me what is exactly the respirocytes will have. So depending on that, we can able to isolate it.
அதுவும் வந்து சாதாரண ஒரு ஆர்பிசி வந்து ஒரு உடம்புக்குள்ள என்ன பெர்ஃபார்ம் பண்ணுமோ அதை வந்து ஆர்டிஃபிஷியலாக பெர்ஃபார்ம் பண்ணக்கூடியது தான் சார். அதுவும் நானோ டெக்னாலஜி தான். அந்த நானோ டெக்னாலஜியில் ரெஸ்பிரேஷனுங்கிறது ஒரு ஆர்டிஃபிஷியல் ரெட் பிளட் செல். எஸ், ஓகே சார், ஓகே சார்.
Circulate ஆக முடியாது இல்லையா? The end purpose is to be doing the job of separation of gases, right? Oxygen or CO2. So after their performance, it can be separated. Okay?
Okay.
Is that clear?
ஓகே சார். என் கொஸ்டின் என்னன்னா ஹைபோதெட்டிக்கல் தியரி அப்படின்னு சொல்றாங்க. நம்மளால முடியுமா இதை வந்து அப்படிங்கிற ஒரு இதுதான்.
Otherwise it may not be proposed. அது உடம்புக்குள்ளே இருக்கிறதுனா it will create adverse effect, right? That's obvious. It will be separated.
To ask something about clinical applications in medicine. So we were telling about only the formation and manufacturing. Started with CTCs, circular tumor cells. In the madri, how can we use it?
Sir, there are several applications we can use. One is, say we have some antibodies that is circulating in the blood. Say for instance, cardiac troponin, a cardiovascular marker, and you have different markers that circulates on the blood. Even you have ctDNA. We have circulating tumor cells, clusters of cell, and we also have several proteins that is present in the blood.
So, in, isolating the CTCs, or how deeply is it helpful?
It's that's what I said. Interaction. What kind of bonding that can take place within the biological marker and your nanoparticles. Say for instance, I have inox nanoparticles. I have a magnetic bead. I coat it on a antibody. So that can actually bind with the cardiac troponin marker, and it can isolate. And we can quantify how much amount of, the markers are present.
Biochemical substance bound, other which we can quantify.
Correct.
It's kind of isolating.
It's mostly isolating, but even if you wanted to have, say for instance, you need to identify now the blood droplet, the droplets that they are creating, they are also infusing cells into it. Now you want to add different amount of drug effluents within one single cell in one droplet. That can also be done. Even like say yesterday sir was talking about the RT lamp, so that is also possible in a microfluidic device. My thing is like we are talking most possibility of how the nanostructures can be made. So sensing is again a different area of interest, wherein you can do several things that can be captured. You can this OPU technique can also be used to identify what is the concentration in the blood the droplets. The interaction, there could be some chemiluminescence aspects that can also be identified.
Right now, in the real situations, are we using this application?
Oh, okay. Right now, it is in research. So we are also working on a lot of, maybe we can collaborate in future to see what possibility that we can do with these devices. Thank you, sir.
Yeah, thank you very much for patiently answering all the questions and also for your excellent talk. Thank you.
Thank you, sir, for such an informative session. Now I request our chairperson, Dr. R. Loganathan, Associate Professor, Cellular and Molecular Theranostics, VIT Vellore, to present a memento to Dr. N. Ashwin Kumar, Assistant Professor of Biomedical Engineering, SRM Institute of Science and Technology, Chennai. Thank you, sir. I would like to call the dignitaries and participants, students and all volunteers to assemble near the stage for a group photo.
இந்த ஒரு நிமிஷம் அதுதான் சார் இருக்கிறேன்